personality disorder, narcissism, and they said they wanted to bring me back as an example for each and every one of those things. I will not be coming back. I'm sorry. I do want to talk a little bit about just some touch-up areas, but I do want to have some questions first before we jump into this. Because we're going to use the whole board. Left side's the bone marrow, right side's the thymus, and then the stuff in the middle is the whole rest of the body, except for this box right here. That's the monitor is in the way of some of the folks, so I'm not going to use that area. Okay. Let's talk a little bit. It's in the handout, but I did not cover it all in a slide or anything like that. Was things like passive versus active immunity or immunization, however you want to look at it. This is something that it really relates to you guys because in the clinic, one of the things that you're going to be doing is administering immunizations to not just pediatric patients, but to adult patients at times. And so when you say passive immunity, we're talking about we can transfer immunity from one individual to another. The best example of that is the mom's antibodies are being transferred across the placenta to the baby. And that's IgG, we talked about that. That's actually transferred over. There are other ones, like when we first started talking about what is humoral immunity. We first learned about B cells because we learned about antibodies before. T cells actually was something that came along in about the 60s and really started understanding what T cells were in the 70s. And yes, I was around back then, but I wasn't an immunologist back then. I was something else. I was a whippet. Right, where's Omega? Radar right there. Okay. So passive immunity at that point meant we started calling it humoral immunity because we could transfer serum from an immunized animal, mouse typically, to another one. So you immunize a mouse, it develops antibodies, collect the serum, transfer it over, and you transfer immunity, at least for a little while, to that other individual. That's passive immunity. Active immunity is the actual immunization that saved the price. And so the vaccinations that you receive, you're inducing the immune response in the actual individual. And another form of active immunization is actual infection. It turns out, in most cases, infection probably does a better job than our immunizations. In the future, we're going to get a lot better at it. We'll be able to induce better immune responses than infection itself. In some cases, we have to do that. And I've mentioned tetanus many times because you can't develop an immune response fast enough to it to help you survive. There's probably some exceptions, but for the most part, you need to have an immunization, an active immunization or a passive immunization against it. So let's talk about rabies. Rabies is caused by a rhabdovirus that, you know what happens. You have a lot of frothing of the mouth and some madness and all this stuff, and then eventually the animal and the person will die from it, so succumb to it. There's a lag phase between getting bit by a rabid animal and before the symptoms start to show up. So there's a time to control that. That is also a passive immunization, and what they do is they immunize you, or they basically transfer antibodies to you to take care of the virus. So you get these injections, and I hear they're pretty painful injections that they have to give you to help you with that. So those are two examples of passive immunization, active immunization, different things that are going on there. And I just want you to be clear about that as you go forward with this. When we talked about humoral immunity, as I said, serum transferred, but with cell-mediated immunity, I've said it about 20 times, T cells have to go to the site of infection. So if T cells are required for an adequate immune response, we can't transfer enough serum to make that work because there's no T cells in there. There are cases where you can transfer cells, and at least in animal models where we transfer the cells and we transfer the immunity, and that's definitely cell-mediated or cellular immunity, and that's why we call those things what we call them. That was just some clarification I wanted to do there. What questions do you have before we get started in this thing up here? Yes?
I have so many, but I'll start with more recent. So in B cell maturation, there's 4 steps to that, but then after that part of the handout, there's some on the H chain and the L going first, the L chain getting the surrogate. When in the 4 steps of B cell maturation does that take place?
Tell me what the 4 stages are, so we're on the same page.
So the rearrangement of the IG.
Okay, that's in the bone marrow.
And bone marrow, and the deletion of the self-recognized.
That's in the bone marrow.
And then the terminal, or the antigen dependent,
Then we leave the bone marrow and go to the secondary lymphoid tissues.
and then differentiation. So
Differentiation.
Where the H and the L chain, at what point is that happening?
Okay. We're going to get into that right here. We've kind of talked about it, but we're going to talk about all the steps in the bone marrow in the order that they go. And that's to any of the, adding the H chain and the L chain together to make an antibody molecule, all that goes on in the bone marrow. Once they get out, that's when you have the somatic hypermutation that we call affinity maturation. So we don't change the H chain and the L chain. They're the same. But it just so happens the DNA that encodes them, that you've already rearranged and is beautiful, we can mutate that during replication. So your original clone doesn't change. Just keep that in mind, because its DNA doesn't change. What happens is it divides. So DNA was replicated, and during that division, during that replication of the DNA, you introduce some additional mutations in the DNA that could make the affinity a little bit better or make it a little bit worse. That's going on in the secondary lymphoid tissues. The actual production of the antibody, once it differentiates into a plasma cell, that can be done right there in the lymph node or spleen, in that secondary lymphoid tissue, and the plasma cell may or may not stay there. It may go to the bone marrow, it may go to the spleen, somewhere where it can produce a lot of antibodies. When it does that, spits out the antibodies, after about 3 days it starts to die. And then if we need more, we just recruit them. Antigen's still around. But I'm going to go through those steps when we're in the bone marrow, and the steps with the T cells in the thymus, and then we'll go to what happens when they leave there. Because here's what we're doing today. We're talking about an infection. We're going to start with a gram-negative bacterial infection. This individual is going to step on something and end up introducing the gram-negative bacteria. And the gram-negative bacteria is going to be like even something as simple as E. coli. But other gram-negatives are going to be like salmonella, which you ingest in food and things like that. But they have a lot of LPS on them, and LPS is one of the most important PAMPs that I told you about. Pathogen-associated molecular pattern. Very inflammatory molecule. And this is the molecule that's really responsible for the sepsis that you see when gram-negative bacteria become more systemic instead of localized in the tissue. Very difficult to treat that because you can kill the bacteria, but the LPS is still there. It's still causing all these inflammatory cytokines like tumor necrosis factor alpha, like IL-6, IL-10, all these things are being produced, and the inflammation is still there. So you have to treat both the infection and what the immune response is doing to the body at the same time. A lot of times what they'll do is they'll give the steroid treatment to knock the inflammation down. Sometimes you see that in a meningitis case as well. All right, it gets into the brain. We don't want inflammation in the brain. It's fine if we have it going on out here, all pus formations, nasty stuff going on, that's fine. But if that's going on in your brain, that can hurt your ability to remember things or even think properly. And that's what happens when you become a provost. You just kind of stop being able to remember things and think clearly. But we're going to talk about infection with a gram-negative bacteria, and then we'll see what the time's like. We'll start with the virus because there's a little bit of difference there that I want you to appreciate. The thing is you think I'm going to start with the infection, but I'm not. And the reason why is because all of the important things that take place irrespective of the infection. All of the stuff to develop the B cells that we need to respond, all of the stuff that we need to make the T cells in the thymus to respond in an adaptive immune response, those take place regardless of whether there's been an infection or not. It's a random process that starts very early in your life, and in some cases, in the fetus, it's already started a little bit, and continues through your life, especially the B cell production in the bone marrow. The T cell production in the thymus, not so much. by the time you get into your 30s, you've probably been exposed to all the different things that you need to, and the thymus will keep just getting smaller and smaller, less T-cell education, less new T-cells. You have a lot of memory T-cells hanging around that will do the job. So, we're going to start, let's start with the bone marrow because that's where the cells are. What's being made there? We have hematopoiesis. I'm not going to write it because I'll spell it wrong. But we'll have the monocytes. And why do I call them monocytes? Because that's what they're born. They're born as monocytes. They leave the bone marrow. And if they move into a tissue, they convert to what we call a macrophage. There's a little bit of phenotype difference to them, and so we call them a macrophage. So right now they're a monocyte. We have the granulocytes, and the only ones we're going to be concerned with right now are the neutrophils. But remember, we have eosinophils and we have basophils that are part of that granulocyte lineage that came out of the myeloid progenitor that's in the bone marrow. And then we also have the lymphoid progenitors, and they are going to make these things that are not even B and T cells, but they're going to be future B and T lymphocytes. They're the lymphocytes. There's also the natural killer cells, and if we get to the virus thing, I'll try to bring up the natural killer cell, but the natural killer cell is kind of like an early version of T cells, but they're not specific. Okay, so they're not really specific for a particular antigen, but they're specific for an altered self cell. So if a self cell gets infected with a virus, some alterations take place on the surface, and an NK cell is basically going around and testing this. Some people call this immunosurveillance. You have cells that move throughout the body, and they're just kind of touching and seeing if everything's okay there. And these NK cells have these proteins that interact with proteins on the self cell, and if some of those are altered or missing, it keys the NK cell to do something. That's when you have some virus, the self cell is going to be killed by the natural killer cell by granulolysis. All right, so what's going to happen is monocytes are going to leave, neutrophils are going to leave, the future B cells are going to hang around, and the future T cells are going to go way over here to the thymus. And stop right there. We'll come back to the T cells a little bit later. What's going on with the B cell? It's not even a B cell. It has nothing on the surface that would indicate to you that it's a B cell. It just looks like a lymphoid progenitor cell in the stem cell. What happens is it starts interacting with the stromal cells in the bone marrow, and there's lots of them. Stromal cells will give it signals through KIT and some other things that we're not going to worry about, but it's basically to keep it alive. And it starts going through the process of rearranging immunoglobulin genes to make a good immunoglobulin. Now, if you can't see what I'm writing down here, let me know and I'll move it somewhere else. Can everybody see right down here? Good enough? All right. So the first thing that happens, we all know that the thing that we're after here is this Y-shaped molecule, two identical heavy chains, two identical light chains. We also know that the heavy chains, heavy chains have a V, a D, and a J segment that makes up the variable domain, but we have lots of choices for each one. The light chain just has V and J, and then there's some constant genes downstream that are also critical for a functional protein. So the first thing to happen in that B cell, that B hopeful, is we have D to J rearrangement in the heavy chain loci. So heavy chain loci, number one. D to J rearrangement in the heavy chain. And just real quick, remember that there are specific sequences that are recognized that mediate that. There's a heptamer, a spacer, a nonamer, and then the other side, the fragment that it's trying to be hooked to, also has a heptamer, a spacer, and an undermer. And we follow that 12-23 rule where there's 23 bases in one spacer and 12 in another, so they match up properly. RAG1 and RAG2 are those genes that make proteins called RAG1 and RAG2 that are involved in this, the recombination activation gene proteins, critical for this type of rearrangement. You only find them in B cells and T cells. Actually, you find them in the things that want to be B cells and T cells. Once they become B and T cells, we don't have to have those around anymore and they get turned off. But D to J happens first. The next step is V upstream of that is going to rearrange DJ. And when that happens, DNA in between each one of these things get looped out. So if we had 40 V genes, V1 through 40 here, and we had the D genes, however many, 23 of those, and the J genes, when this rearrangement takes place, some of this DNA is lost. And then when this rearranges down here, some of this DNA is lost, and the promoter upstream of the V gene segment now is close proximity to an enhancer down here. And I'm going to erase that here in a minute. So now we have something that we can actually transcribe through and maybe make a functional protein. Maybe. Maybe we'll make a functional protein. But we have a caveat here. Heavy chain can't go to the surface without a light chain. In order to go to the surface, you have to have heavy plus light plus those signaling molecules, Ig alpha and Ig beta. Remember, they have the ITAMs on them that helps actually do the signaling. They're associated very well, very much so with the heavy and light. We have this, but we don't have that. So the next step is for a surrogate light chain to interact with the heavy chain. That associates with Ig alpha and Ig beta and goes to the surface. All right, so it looks like a pre-B cell receptor, what we would call a B cell receptor, except it doesn't have a real light chain. It has a surrogate light chain on there. What happens at that point is the stromal cells that are interacting with it give it a signal that you've made a functional heavy chain. So the signal goes down to the nucleus, turn off RAG genes, start proliferating. There's one clone that we're talking about. We're just talking about one clone right now, but this is going on with millions of B cells in the bone marrow during the day. That one clone then goes through 5 or 6 rounds of replication, and we have a bunch of clones. Next, what thing is going to happen, once we've gone through that rearrangement, then we're going to go back and we're going to start rearranging the light chain. And we have to only have to do the V to J rearrangement. And so once that happens, then the light chain associates with the heavy chain. We finally get the H plus the L plus the Ig alpha and the Ig beta, and we have what looks like a functional, real B cell receptor. It goes to the surface, and then we have to make sure that it's going to work and it's going to not be self-reactive. So we went to a positive selection state without the light chain. Basically, we've got a functional heavy chain, and so we selected those and said, you've got a good heavy chain, make some more of you, we have some clones. Then we go back and we make a light chain, and we have to make sure, well, this light chain in connection with this heavy chain might be self-reactive. And remember, so far with the heavy chain, we developed, I'll use this one, we developed those 3 CDRs by the VDJ rearrangement. Never really kind of came out and made that clear, but the 3 CDRs that are the 3 loops that stick out are in that VDJ part of the variable domain. So by rearranging these things, you actually produce those 3 CDRs, and then we also hypermutate them a little bit later. So at start, we just have the heavy chain. We add the light chain. Now we have 6 CDRs on this arm and 6 on this arm. And remember, this is an FAB fragment. by itself. One arm is an FAB, one arm is an FAB. This part down here is the FC. But if you cut the FC off and these two are together, it's called an FAB prime 2. So if I was to ask you how many CDRs are there in an FAB fragment, how many's here? 6. How many's in a full antibody molecule? 12. Okay. So, and we have a valency here. Now, if we're talking about a full produced IgM antibody, then you really have 10 because you have 5 of these that are hooked together. But we'll worry about that later. So, we have no infection going on. We don't even care what's going on outside the body. So, a completely random process taking place in the bone marrow. The B cell has rearranged the heavy chain, found out that it's functional, rearranged the light chain, put it up on the surface, and we turn off RAG1 and RAG2, and we have this B cell receptor that then has to be tested to make sure it's not self-reactive. And in the slides that I gave you, there was one particular figure where it says if the B cell in the bone marrow has interacts with a self-antigen, with a very strong interaction with the self, then it can really send it into anergy. It can actually go into apoptosis, not anergy. It really actually kills the cell before it gets out. Millions of cells die in the bone marrow every day. Every day. This process is going on, and they're not going to be functional cells, so they get deleted. Yes?
So at this point, is the antibody already specific for an antigen before you've encountered it? Okay. So all these, you just are creating a bunch of millions of
of different B cells.
Mm-hmm.
But that B cell is specific for one thing, because of the CDRs that it has. And outside of those 6 loops out that are sticking out there, every one of those B cells will look exactly the same. Because they all have IgM. They might look a little bit different because they might have a kappa light chain versus a lambda light chain. And that's just an additional diversity that's added. But yes, that B cell, before it leaves the bone marrow, is already antigen specific. And it's pretty good specificity, but we can make it better once we get out. So the B cell is antigen specific, it's just never seen its antigen. And this is why I say that what goes on in the bone marrow is antigen independent. But there are self-antigens involved. So we can just say it's foreign antigen independent because no foreign antigen is involved in the development of the B cells. They don't run into the antigen until they get out and get into the periphery. All right, so B cell now has an antibody on the surface, and we'll make it look like this. Here's the antibody on the surface. One of the critical things that I want you to remember is that when we were looking at the VDJ, do you remember what the first constant gene segment was that we run into? Yeah, mu. That's right. It was mu. Mu is the first, and delta was right beside it. So when we transcribe through we have a VDJ, and then right downstream is going to be the mu. There might be some stuff in between here, but mu, and then we have delta. We're going to transcribe from the promoter this way, and we will produce a VDJ, and then the constant gene segments of mu will be in the transcript and delta. And so the first thing to go up on the surface of the B cell when it's coming out of the bone marrow, every B cell coming out of the bone marrow has IgM on its surface and IgD of the exact same specificity. Because we haven't got to the point where we're working with the ones that are downstream. All the gammas and the alphas and the epsilons and all those are downstream. So we have an IgM, IgD positive B cell that has left the bone marrow, completely naive, never seen its antigen before in its life. And it then goes to all of the different secondary lymphoid tissues, lymph nodes, spleen, and malt, gall, all those different places that has what are loose lymphoid tissues. I was reading something, they're finding even newer patches where there's lymphoid tissue hanging out in the body that were really hard to find before. So there's immune responses taking place in lots of different places throughout the body. So, key thing here is the natural effector cells, monocytes, they get in the periphery, go get into the tissues, and are hanging out looking for antigen. Some of these are also, remember the dendritic cells, the DCs, are also going out, and they're hanging out in lymph nodes, in tissues, all over. They're just going wherever they're needed. And in the granulocytes, most of the neutrophils, the most prevalent, most numerous white blood cell in your circulation. Neutrophils can be in the tissues, but typically they're just floating through the blood looking for a place to go. They're not involved in antigen presentation. They're not involved in that stuff. They're involved in killing stuff. They're just kind of a hired killer. Yes?
Go back to the bone marrow. I don't understand how, or where are those proteins that are teaching them that they are antigens? And how can you have all of them in the bone marrow that somebody is going to encounter in order to make B cells against them?
You don't. And that's exact. So there you don't have, so when we get to the thymus, we talk about MHC for self-peptide and all that stuff. The MHC molecules come up with surface. In the bone marrow, these stromal cells have a lot of things on their surface. And basically the idea is, you know, with the antibody molecule, when it's going through what we call positive selection, the idea is, can this molecule, this IgM, hooked to a surrogate light chain, cause a signal if it touches something? And to tell you the truth, the way I understand it is the surrogate light chain gives it a little bit of flexibility in what it can bind to, is the way I understand it. But that's something that I haven't really learned much about, and I don't understand the nuances of how that happens, because as Dr. Latazzi said, there's no way, well, they're not recognizing a specific antigen. We're just making sure they don't recognize self-antigen when they have the full receptor on there. But there's some mechanism in the stromal cells that are in the bone marrow that are interacting with the B cells that allow the B cell to be signaled that we have a functional heavy chain molecule. Now go get a light chain and we'll check you out again later during negative selection. Then it's if you bind really tightly to a self-antigen, then you do get a signal. So there's a lot of unknowns going on. There's a big black box inside that B cell that would say, okay, what's the difference between signaling through IgM and the surrogate light chain and the full B cell receptor, positive selection versus negative selection? Don't know. We just have to believe the people that tell us these things and move on and try to figure it out as we go. So, yeah, it is kind of mind-blowing when you hear this and you just go, oh, that's just crazy. That can't happen. Well, it happens. We just don't know how it happens. But for our purposes, because all you guys care about is getting the question right on the exam, right? The positive selection is just this heavy chain with a surrogate light chain, and its interaction with its environment in the bone marrow says the heavy chain seems to work fine. Go make more of yourself, proliferate, then go make a light chain, and let's see if you're still a good antibody or a bad antibody, a good B cell or a bad B cell. And then they leave. Once they get past that, they leave and go out into the periphery. They are going to float to the lymphoid tissues looking for their antigen, like look through the lymph nodes, the spleens, those types of things. And then we'll kind of switch again now to the thymus. Still not talking about infection. Still not talking about antigen. We don't care about antigen at this point. But what has happened now is the thing, this lymphoid progenitor that thinks it wants to be a T cell, and again, I have no idea how this happens, if it's a random process or if there's certain of these lymphoid progenitors that decide I'm leaving the bone marrow and going to the thymus. Not sure what's going on there, although there are a few markers that are found on those early cells in the thymus that would indicate that there may have been something that kind of told the guy to leave, told the T cell to go to the thymus. What color are we going to go with here? We'll go with white. Okay, so T cell has we're just going to go with the alpha and the beta. because gamma and delta will work in there, but remember that these are kind of two fundamentally different types of T cells. Alpha and beta T cell receptor positive cells are going to recognize CD4, are going to be CD4 restricted or CD8 restricted to class 2 MHC and class 1. I think you ought to have that down by now because it's been said about 20 times and it's critical. It's an important thing to understand. So we're going to focus on the alpha and the beta T cell receptor. It turns out that these two chains work a lot like what we saw with the immunoglobulin heavy chain and light chain. What's going to happen first is we have the beta chain has V, D, and J segments. The alpha chain has V and J. And it turns out that the delta locus is right there in the middle of the V to J segments. And when that rearranges, it loops out, but we'll get to it later. So the first thing that happened is the beta chain, not the V, but the DJ rearranges. And so those two rearrange. Again, RAG1 and RAG2 proteins are binding to those recombination signal sequences, bringing them together. And I didn't get into the junctional diversity, but remember that there's imprecise joining of the D to the J, the V to the DJ, and the V to the J in the light chains and in the alpha chains. That imprecise stuff can add diversity, or it could make it a non-functional protein. So D to J happens first. Then we have V to DJ rearrangement. And just like we saw with the immunoglobulin heavy chain, the T cell receptor requires, requires CD3 molecules. And you remember what they were? They were naturally Greek letters, gamma, delta, 2 epsilons, and 2 zeta molecules with the ITAMs, the immunoreceptor tyrosine activation motifs that actually do the signaling. T cell receptor can't go into the surface without CD3. It won't get packaged, it won't go up on the surface, so it's a complex. T cell receptor is always going to be with CD3. But right now, what do we have? We have a beta chain. So this beta chain will interact with the surrogate alpha chain and associate with CD3 and go to the surface and make sure that everything's working okay. All right. And what I will say about, remember the gamma delta, so we also have the gamma delta, it's kind of a race. The same time the beta starts rearranging, both gamma and delta start rearranging. So you could get a gamma delta T cell coming out of it at multiple stages during the development of an alpha beta. It's doing all this at the same time, regardless of antigen. Doesn't care what the antigen is, it's just doing it in a random process. So this goes up on the surface. We find that we have a functional beta chain by interacting with the thymic epithelial cells that are in there. And then we kind of do the same thing that we did at the B cell.